So the thing what I'm going to talk today is, okay, so that's my disclosure. So that's, is about the two major hurdles that we faced when we, in my lab, changed from a basic stem cell biology lab about 7-8 years ago to a translational, and started to actually prove that yes, it is really true that we always said about stem cells before when we did just basic pluripotent stem cell research that these cells hold promise. We've always said that, but we never thought about actually doing anything about it, so that how we actually prove that it is really true. And about 7-8 years ago, so that we started thinking translational and identified the two major hurdles that we thought that has to be solved in order to move cell therapy into medicine. And these are the safety. We have to make sure that we will not create harm by putting cells into a human being, cultured in vitro. And also we've heard a lot up to now that how important it is to actually develop something which will allow us to use allogeneic cells to treat disease. And so these are the two things that I'm going to talk about. So that there's two platform technologies developed in the lab. First, I just wanted to show that our motivation and vision here, that if you have solution for these two major hurdles, you may be able to do this. So that practically I have let's say one safe cell line, or maybe two, a female and a male, and as a universal cell, which could be the source of off-the-shelf cell products serving all humankind. So and then first, these two more or less published, these two platform technologies, but I'm going to through those which I think it is very important and tell these two technologies different from the other attempts to solve these problems. So one is the safety issue, which is published already 3 years ago in Nature, where we came up with a solution so that how to ensure that the cells are not going to create tumors if they are implanted into a human being by placing a kill switch into a very special genomic position where we can ensure that the cell, using most of its dirty tricks to knock out something or eliminate something or delete something like a kill switch from the genome, will have a hard time to do that. And this safety system, which we called a suicide gene or a kill switch, was knocked into a cell division essential gene, into a 3' UTR of the gene, creating a bicistronic message with a cell division essential gene product and also the kill switch. So and then we realized that if we do this, that probably we give the suicide switch, kill switch a very, very high level of protection, because if the cell is trying to eliminate that, then it's going to eliminate also that product or gene product which is needed for cell division. And we have to do this obviously in a homozygous manner, because one of the most prevalent dirty trick of cells to get rid of something from the genome is loss of heterozygosity. So if you have a homozygous way that we can protect also or eliminate that trick of the cells as well. So we looked at the genome and the cell and the genes which is required for cell division and identified quite a few, and we took site-dependent kinase 1 and TOP2A as a prototype to make this link between the kill switch and these genes, and we took the HSV-TK as a kill switch which can kill cells in the S phase with a prodrug against cyclovir. So, but we have to also understand for cell therapy that nothing absolute safety in our life. Whatever we do, so that riding a bicycle, flying here on an airplane, so that we take a risk. So it's also true for this kind of kill switch. It's not absolute safe, so that there is a way still remained for the cells to inactivate such a kind of thing. So and then this is what, wait a second, yeah. This is what I want to show you here, so that now we had this link between the cell division essential gene and the suicide switch, and then you start growing up the cells, and you realize that the cells still remain some tricks, for example, point mutations. So the point mutation could eliminate the catalytic domain of this kinase, so that practically could create a killing of the kill switch, and the cell is still able to divide. So, and then when this slide practically now show, let me see, that when the cell divides, so that the cell, every division, the cell has the possibility that to mutate the suicide gene, but keeping the CDL, the cell division essential gene, functional, or by eliminating all of them, but because it's, so these are the possibilities of an outcome of cell division. And then we were lucky to be able to calculate these possibilities, estimate these possibilities, and practically model what's happening if you start growing up the cells. From a single fail-safe cell, homozygous for this modification, you start with one cell and then start creating a pool of cells or a batch of cells needed for therapy. And this is what we call the genotype transition matrix when you grow up cells, having this kill switch embedded into cell division essential gene. So it's quite complicated, so that we had to do practically lots of computing and Monte Carlo modeling to actually establish what's the risk of, even with this very, very smart, sorry about this word, placing the kill switch into the genome, we still could have escapee from this very, very tight holding of or keeping the cells safe. Because if the cell managed to mutate on both alleles of the kill switch, so practically this is what we call the escapee, could escape from ganciclovir treatment if these cells are getting tumorigenic. So so then after that we started thinking about how we can actually tell a patient what does it mean, how much risk you take. And then this is what we came up with, a simple definition of the fail-safe level. That the fail-safe level is a number which tells that the odds of picking a non-fail-safe population for treatment is 1 out of this number. So practically I can tell a patient who is waiting for cells in the eye to treat blindness that because you need, let's say, 100,000 cells, for example, then the safety level of this treatment is 1 out of, let's say, 10,000, which means that 1 out of 10,000 people who is getting this treatment could get a batch of cells for the treatment which is not fail-safe, which could have escapees in it. So, and then with this definition, we started calculating the fail-safe level in the function of cell therapy needed to treat disease. And this is which we came up with, so that, and it's one of the figures in the paper, which practically shows that if we create a homozygous knock-in into one cell division essential gene, which is CDK1, and this is the cell number, log10 cell number needed for cell therapy. This is 10 to the 5 to 10 to the 10. 10 to the 5 is the number that the people who are working or trying to treat blindness is thinking about, so that 100,000 cells, and 10 to the 10, which is 10 billion cells in the mind of people who are trying to treat cardiac infarct. This is a number of cells needed to inject into an infarcted heart, 10 billion. And then we started calculating the fail-safe level, and then showed that for eye treatment to 10 to the 5 cells, fail-safe level is 10 to the 4. So I can tell that the risk what you are taking, that not all the cells are going to be fail-safe if you get 10 to the 5 cells into your eye, is 10,000, which 1 out of 10,000 batches is going to be non-fail-safe. But the bad news is that if you're thinking about diabetes, which is needed 10 to the 9 cells, the fail-safe level practically goes down to 0. All the batches of 1 billion cells is going to contain escapees from the kill switch. So that's why, so that in the paper, so that we just suggested, no, okay, we can do it with 1 gene. Why don't we do this with 2 genes, CDK1 and TOP2A? And if you see that in this graph, that if you modify two genes with homozygous manner with this kill switch, the fail-safe level goes up astronomical. So, for example, for the heart, which is 10 billion cells, the fail-safe level goes up to 10 to the 6. Only 1 out of a billion, 1 out of a million cardiac infarction is going to get a non-fail-safe batch, which could contain escapees. And it is so much that I even showed that how many cells a human being has. And this is 3.7 times 10 to the 13 cells we have in our body. So if you think about 3D printing a whole human being with in vitro culture cells, you can actually print 1,000 people, and only one of them is going to be a cell in the body, which is not fail-safe. It could escape from the kill switch, right? So anyway, so that we showed that, yeah, it is working, so that we went into the mouse embryonic stem cells and created cancer, created teratomas in the mouse, injected this fail-safe mouse CSL cell subcutaneously under the skin, and then let it develop into a teratoma, which eventually, if you don't do anything, is going to kill the mouse because it's growing really big. And then using the ganciclovir, which is the prodrug of the kill switch, at the size of 500 cubic millimeters, we could actually kill all the dividing component of the teratoma, leaving the undivided, already differentiated, mitotically arrested component of the teratoma, stabilize it. We put with a short ganciclovir treatment this tumor into a dormancy. And it stays there, didn't disappear, it stayed under the skin for a really long time. So that's for the life of the mouse, which was good, showing that, yeah, so we have a solution for safety. We also repeated this experiment with cancer cell lines, like a breast cancer cell line, and then showed that we can actually control breast cancer development by using the fail-safe system. So when you have a solution for safety, you can start thinking about systems or ideas which are intrinsically dangerous thing to do. And this is immunocloaking, hiding the cells from the immune system. And this is what I just quickly actually go through, because it's more or less published, so that there's a bioarchive manuscript now posted, and also there's a review that we wrote 2 years ago, and describing practically the efforts, what others are also doing, and we just heard what we are trying to do and others trying to do now is creating hypoimmunogenic cells, or maybe immunocloaked cells. So we did it differently, so that we thought that we are not going to worry about the HLA mismatches. We want to actually generate a system which going to work across mismatch. And this is where we started learning immunology, which was painful but very exciting. So we identified 8 immune modulatory genes, which we thought that it is necessary to express by the cells. Most of them are cell surface, because we wanted to have a local action, a local immune suppression, or not immune suppression, but local cloaking of the cells. And then these are the genes which are now, we picked CCL21, PD-L1, FAST ligand, SERPIN, HLA-G, CD47, MFGE8, and CD200. These are all involved in many different systems in hiding the cells from the immune system, like cancers are using it, the placenta is using them. So, and then we decided to overexpress this gene in the cells, and then, and it worked. So it was beautiful to learn that if you take black 6 mouse CS cells here, transplanted the black 6 mouse CS cells into the black 6 mouse, which is allogeneic, no surprise, these cells are staying there, not rejected. But if you take a fail-safe but not cloaked cells and put it into an allogeneic recipient, they very rapidly reject it. So even in day 5, most of the mice cleared up the allogenic cells uncloaked. Now we do it with cloaked, with the B6 cells expressing these 8 immunomodulatory genes, and then putting into an allogenic recipient like in FVB, so they stay. It's a short experiment, because day 17 and the tumors start growing, So we had to combine, in fact, now the fail-safe technology with the immune cloak technology to have some information about the long-term acceptance, right? You remember that slide which actually shows that we can stabilize the teratoma and put it into dormancy under the skin. So now we have the two systems together, and we repeated this experiment and stopping the teratoma growth at the size which is still acceptable for an animal facility, and it led the mouse to go for years and see what's happening with the allogeneic tumor. When it is fail-safe, which is needed to put the cells, put the tumor into dormancy, but also expressing these 8 transgenes. And when you see that when we apply ganciclovir treatment in this mouse, that we can stabilize these tumors under the skin, and then we have all a year, and they are staying there, staying under the skin without being rejected. So that practically this is the evidence for long-term allogeneic tolerance without the need of systemic immunosuppression. Cool thing, we didn't expect that these tumors are also immune privileged, like our brain or our eye or testis, because when you create this immunocloaked allogeneic tumors under the skin, and then we put human embryonic stem cells inside, which is luciferase expressing, they are protected by them, so that even the xenogenic cells, Y-type genes and xenogenic cells are protected by this tissue, which is now immunocloaked and fail-safe. Just to show you that how the teratoma looks like when the human cells are inside. So this is like an organic capsule, which is fail-safe and protecting the xenogenic cells in an animal. So I just quickly show you a few things, so that this is what we have, a solution for fail-safe and allograft cloaking. If you touch the genome, you start thinking, okay, why don't we go a little bit further and then further modify the genome and also use these cells to express biologics, which could synergize with the cell therapy effect and making the cells even better therapeutic than a Y-type cells. And this is where we started thinking about also designing biologics, so created a sticky trap for VEGF and DICO receptors and endogenous factor expression, and practically use the same idea, fail-safe, immunocloaked, and producing something, secreting something to several disease models like blindness, arthritis, diabetes, hypoparathyroidism, aging, chronic pain, multiple sclerosis, and COVID-19, the latest one. All requires specific biologics to be expressed to synergize with the cell therapy or being just a therapeutic effect. VEGF, anti-VEGF works in treating eye disease, so that practically putting retinal pigmented epithelium, expressing an anti-VEGF into the subretinal region of the retina and using laser-induced ablation model of the retina and looking at the neovascularization after that. If we inject these cells into the eye, we can suppress the neovascularization of the retina, which is practically the underlying problem with, for example, age-related macular degeneration. So same thing also what we are using or doing with multiple sclerosis. We generated an anti-GM-CSF trap and showed the effect if we are creating a source of anti-GSF in an animal model of multiple sclerosis, the EAE model, and looking at the clinical scores. And this is what when there's a transplant expressing anti-GM-CSF and the EAE score showing a therapeutic effect of this implant to the disease. The last thing what I want to show you, which is not a big surprise, but you are obviously interested in brain, and we also tested, and we were brave enough with the safe cells and also immunocloaking to put pluripotent cells into the brain, which everybody is afraid of. It is going to create a teratoma, and this is what we don't want, obviously. Now, with having the fail-safe and also immunocloaking, we started putting cells into the brain, pluripotent cells, and looking at whether we can actually use the kill switch to actually make this whole thing, which is total crazy, safe. Right? And this is an experiment when you put embryonic stem cells into the brain and you see that it's growing and it's going to kill the animal eventually. And this was the experiment with the same number of cells put it into the brain, and we applied ganciclovir treatment right after the transplantation. And it shows that practically it eliminates the graft, a kill switch prodrug, when the cells are put into the brain straight. And here was what's quite interesting, because now we delay the ganciclovir treatment, let the embryonic stem cells in the brain to differentiate into a tissue, which is a teratoma growing. But before any harmful effect, we applied the ganciclovir treatment and stabilized the tissue which is generated in the brain from pluripotent cells, which are differentiated and not dividing. So we actually now rename this tissue, and don't call it dormant teratoma. We call this is a friendly tissue in the brain, right? That you can use for many, many good things to do, therapeutic things to do. So, but the disease is tricky in the brain, because it's true that we can put into the cells, even if it is allogenic, so that's not rejected because the brain is immune privileged, right? So we can put allogenic cells into the brain and not reject it. But if we induce disease, for example, EAE in the brain, that the immune privilegedness is not working. So we have these cells rejected when we induce EAE, for example, in the multiple sclerosis model. So therefore, in order to protect our allogenic transplant in the brain in a disease situation, we also have to use immunocloaking. And if you use immunocloaking, then you can protect practically the allogenic cells, even in a disease brain situation, not being rejected. So I want to close to this with this one, so that I think that having this solution for these two problems in the mouse, so we are in the position of really dreaming, or not even dreaming, so that planning to get a source of pluripotent cells, which could be the source for therapeutic cells off the shelf available to treat human disease. One thing is that now I showed you only the mouse data that we have. Obviously, we are interested in a human. So all these 8 genes has human orthologs and functional orthologs, and generating obviously human cells. The testing of immunocloaking in human is obviously not as easy as in the mouse, but it is going on, and it seems to us all the in vitro studies that we performed, these cloaked and fail-safe human cells, showing what we expect from the function of these immune modulator genes, how they affect, for example, immune cells of the recipient or the host when they meet with a total functioning human immune system. So the most important slide is practically all those who actually work with these two technologies, the Toronto lab, which is this one, and the company, which is a spin-off company from the lab. And we have a collaborator at the university, which is a phage display facility, providing us also lots of biologics. I have a very small lab at the Monash University in Army, and also developing a massive collaboration also in Hungary with these institutions here. So thank you very much.
Thank you. Maybe I'll just take the privilege of asking the first question, since there's people lining up to ask later ones. I noticed when you had the animal experiments where you were treating with ganciclovir in the teratoma model, I think it was, there were some later relapses, including those which were then treated by subsequent ganciclovir, and which were sensitive to ganciclovir. So the cells that were seeding that relapse were effectively ones that had escaped, not by mutation because they were still sensitive, but perhaps because they were quiescent at the time and weren't affected. So what's the risk of heterogeneity in the cells leading to an escape profile later on? And a sort of follow-up from that really is, may that be modified by selection? in your modeling studies, you've had a sort of selection-free environment. But in the human population, I suffer from cold sores. My response to that is I go and get acyclovir and dose myself up with acyclovir. So that may impose a selective pressure on these populations of cells that may promote a faster rate of escape variance, if you like, than you might see in a non-selected population.
Yeah. So with the teratoma-size, there are several things which is changing it. For example, cyst development. And then when you're just measuring when popping the teratoma-size, it creates a variation in this one. So that's one. But you are right. So there's a possibility that after a brief ganciclovir treatment, you're not killing everything which is capable of divide. You are killing the fast dividing cells because the ganciclovir kills only in the S phase cells. So if this is a sleeping beauty type of cell, which actually doesn't go to the S phase during the ganciclovir treatment, could wake up, right? So that's why it's important with this failsafe to monitor also, so that just to make sure that there's no sleeping cell in that, right? And then absolutely essential to have a system where you can monitor, because these cells, even if they went into a mitotic arrest, could have a cancer development in it, or cancer potential, a mutation which could re-kick this whole thing. But this is when the failsafe system can also, just an additional ganciclovir treatment, eliminate the later runaways. Important to monitor, no question, but this is our own cells also, so that we have to monitor our own body in order to identify if something wrong starts happening, right? Yeah.
Questions from the floor, at the back first.
Thank you, Andras. Was it about your, what you called friendly tissues in the brain as a means to deliver therapeutics? Are you not concerned because there'll be a heterogeneous population of cells that may, in of itself, secrete something that has a biological effect on the brain? So how are you thinking about avoiding that?
Yeah. So I mean, you're right. So this is a proof of principle experiment in the mouse, and that we really just wanted to know whether we can put cells, pluripotent cells into it. If you want to work forward therapy, obviously we can use more defined tissue than a teratoma, which is a total random thing, right? But then we have candidates there, like we can put, for example, retinal pigmented epithelium into the brain as a very stable, very robust cell, not dying. And then we have some experiments now showing that if you are going toward the clinical thinking, we are going to select the cells which we are capable to make from iPS cells, and it's going to be much more defined. And then we can test also what else these cells are creating and make sure that it's not going to generate problem, what you thought, yeah.
Hi, thank you very much for an incredible talk. I was wondering, have you tested your cells in either humanized rodent models, or at least, say, the pig model, since rodents aren't, the immune systems of rodents and humans aren't too comparable?
Yeah.
But say, moving into the pig model and really testing this technology first would be really informative.
Yeah, so this system, so that it's designed in a way that we can protect allogeneic cells, right? And then, so that's why we didn't really try actually into a pig model unless the pig is humanized or mouse-sized. So that's one answer to that. And the other is there's no real good humanized mouse, so that we don't believe that we can recapitulate the proper functional human immune system. Is that there are people are doing it, so we try to avoid, so that we are just putting together in vitro testing, like co-culturing with PBMC, and then see how these cells are changing the PBMC composition. And it looks like that they are doing the same thing, both in the mouse and the human system with the PBMC. So likely it is going to work also in human. Next step is to use a non-human primate model. And then we might have access now to a marmoset system. We have a marmoset iPS cell line, fail-safe, immunocloaked, And we are at the point that we are testing it in the marmoset.
Thanks for this amazing data. I have just a question, thinking about the logistics of transferring this into a GMP setting. Could you just clarify, and I'm sorry if I missed this, how many genes in total were gene edited in the IACT cells, and for the overexpressing genes, are these all placed into a safe hardware?
Yeah. So the fail thing is targeting, homozygous targeting. We have to use CRISPR, maybe not necessarily the CRISPR-9, which is enormously expensive, but maybe another CRISPR type of targeting, which is not as bad as license-wise CRISPR. So that's one modification, which is not too difficult with the CRISPR because we are frequently getting homozygous knock-ins in the cells. So the immunocloaking, as I said, 8 transgenes, right? So there's 2 ways to go. So that for being clinically acceptable, so that we try to reduce the number. And the way how we're reducing it, we're creating polycistronic messaging, or the transgenes, and also putting them into a defined genomic site. And this is an ongoing project, just what we are doing now in Hungary. We are targeting highly expressed genes, right, with the immune modulatory genes, and then just 3 is needed, so that it's relatively not too much manipulation, but certainly it's a manipulation, especially when you're on the top of it, you want to introduce your expression system for the biologics and want to do it in an inducible manner, because it's many times you want to have this inducible. You don't want to express constantly things, that only when it is needed. Yeah.
And then we have one online question from Dmitry Ovchinnikov. Great talk as always, Andras. What could be the prospects of tackling the cloaking transgene silencing? And are we ever going to be able to find an unsilenceable locus for overexpression in the human system? Also, what are the best alternatives to TK ganciclovir?
So, I mean, first, what we're trying now to with the cloaking genes is instead of going random integration, we are going to a defined site. And then practically the idea is that where to find a highly expressed site in the genome. And the approach is that let's see expression levels of housekeeping genes and use those housekeeping genes, which is really highly expressed, and then kind of piggyback or knock in our things in the 3' UTR of this one. So that's one. But what was the ganciclovir issue?
What are the best alternatives to TK ganciclovirs?
Yeah, so the TK is in a way good because it eliminates only the dividing cells. So in many situations, you don't want to eliminate this whole implant, right, if something is happening. You want to eliminate also just the runaway part of the implant, right? So that's I think I do believe that this is going to be a good thing for many implications. Also, you can see that maybe there is a possibility also where you need for removing the whole implant. You don't want this to be there forever, right? And this is where we are developing also so-called painkillers, which kills the implant regardless of the stage of proliferation.
Any further questions? If not, thank you.